Okay, so welcome to this video. In this video, what we're going to discuss is personalised cancer medicine. So let me outline the structure then for this video. What we're going to start off with is a discussion of the carcinogenic process. Okay, so we're going to discuss a very basic model for how cancer develops. Okay, that's going to be essential for us to be able to understand anything about cancer medicine. Okay, so we're going to start off with a basic model of carcinogenesis. Then what we will do is we will draw attention to the concepts of inter-tumor heterogeneity and intra-tumor heterogeneity, because both of those are going to be quite important when we discuss personalised cancer medicine. Okay, then what we will do is we'll actually define what personalised cancer medicine is, and we'll contrast it to classical chemotherapy. Okay, we'll then define a few related terms. So we'll look at the terms pharmacogenomics, pharmacogenetics, and also targeted cancer therapy, which are all very much so related to the concept of personalised cancer medicine. Okay, then what we'll do is we'll discuss a little bit of background genetics, okay, which is going to be needed for us to understand personalised cancer medicine, and then we'll go through some examples of personalised cancer medicine as it stands at the moment. And it's fair to say that at the moment, personalised cancer medicine is in a very primitive form. Okay, we have a few very basic examples of where personalised cancer medicine is starting to be achieved, but really personalised cancer medicine and personalised medicine more generally, because it goes beyond just cancer therapy, it's a aspiration rather than something that has actually been achieved yet, and in 100 years' time this will be far more developed than it is now. Okay, right, so to begin then, we're going to have a basic discussion of the process of carcinogenesis, okay, which is the development of cancer. Okay, right. So basically, our theory of how cancer develops is that you start off with some cell in some portion of the body. Okay, so maybe it could be a cell that lines a bronchus in the lungs, or a cell that lines the gastrointestinal tract, or a cell of the liver potentially, etc. So some cell basically. So I'll draw this here. And originally it was one of the normal cells of the body. Okay, so here it is. Here is its nucleus. And originally in its nucleus it had those 46 chromosomes, and those 46 chromosomes had the normal genome of that person. Okay, so it had the germline genome. Right, so the idea then is how cancer occurs is it starts off by one cell getting a mutation in a gene that then allows this cell to overdivide. Okay, so potentially you get overactivation of some gene that promotes cell division. Okay, or you might get some loss of function mutation in a gene which stops the cell from dividing. Okay, so what's going to happen then is this cell is going to over-divide. Okay, so what's going to happen is it's going to copy itself multiple times, like so. So you're going to get loads and loads of these cells now. Okay, and they're now being drawn much smaller than originally. Okay, so here is our original cell here, in red, and that's made loads of identical copies of itself. Now, an important point is that if this mutation that this original cell got is going to actually lead to the development of cancer, then it has to be heritable, okay, i.e. when it divides, when this red cell here divides to produce two new cells, both of those cells now have to have that same mutation. If the mutation was not heritable, then it's not going to trigger cancer. So you need some heritable change, basically. Okay, so what then happens is that this red cell here produces a whole clone, as it's called, of cells that have all got this same mutation that causes them to overdivide. So effectively you produce a whole population of cells which are all genetically identical and which are all over-proliferating. Okay, right. Now that's not cancer yet. Okay, this, if it was actually visible, if this patch of cells that you've produced, which are all over dividing, is actually forming a visible, palpatable disturbance, then we might call this a benign tumour. Okay, so at the moment it's not cancer. Benign tumours are not cancer. They're on their way to becoming cancerous, but they're only part of the way through. So this at the moment is a benign tumour made up of this clone of these cells which are all over dividing. Okay, now this is where I'm going to simplify it completely down. Okay, I'm going to make this model as simple as it can possibly be, and I'm going to say that carcinogenesis, we can model it as being a two-step process. In reality, it is not a two-step process. But what I'm going to say is I'm going to simplify it down to now saying that one of these cells within this clone of cells which are all over proliferating is then going to get another mutation. Okay, so you need one cell in this massive great clone that you now have of cells which are all over dividing to get another mutation. And this mutation is going to make it motile. Okay, so some mutation that's going to make it motile. Okay, now what do I mean by that? Well, normally cells in your body are not moving relative to the body. Okay, so they have their little positions in the body and they stay there. Okay, they do not move around. Okay, and initially these cells in this benign tumour, even though they're over dividing, they are not motile. They still have this property that they stay put. They stay in their little position. Okay, but now what's going to happen is one of these cells in this clone is going to get some mutation that now allows it to move around. Okay, and what this then causes is that this cell that has got this mutation that now is going to allow it to move around, it also has the mutation that means that it overdivides. So what you're going to then produce is a whole clone of cells which are all overdividing and all motile. Okay, so let me show this. We'll highlight these ones that have both mutations now, both the overdivision mutation and the motile mutation in orange here. Now, of course, we'll still have all the red ones that are just overdividing, so I'll put these on here. Okay, like so. So these are all these red ones. These are the cells that only have the mutation that means that you overdivide, but not the mutation that means that you are motile. Okay, so one cell in this original red clone that got this mutation that meant that it was then able to move around. Now, because it has the mutation already that means that it overdivides, it's now going to make a whole little clone of cells that are identical to it, which is the orange clone of cells, which are now all overdividing and all motile. And the idea is now that these are going to start moving off into the peripheral tissue. Okay, so they're going to come invasive. Okay, they're going to start invading the surrounding tissue. So if this was happening in the liver, for instance, this orange clone of cells is now going to start invading the healthy liver tissue, the liver cells that don't have any mutations in, and that's going to start causing problems for those healthy cells. Okay, so they'll start invading the healthy tissue and causing problems there. In addition, the dangerous thing and how cancer generally kills you is what happens is these invasive cells, these cells that are moving around, but sooner or later they will bump into a blood vessel. Okay, so let's say this is a blood vessel here, which I'll colour in. I won't colour it in red because we've already got the red clone of cells, but I'll colour it in purple here. Okay, and what's going to happen is these orange cells are going to start invading into the blood vessel, and then they're going to end up in the blood. Okay, so you'll get cancer cells in the blood. And then these orange cells, and I should have said that actually, when you've got this final orange clone of cells here, this is what's known as a malignant tumour. Okay, so the orange cells, the orange clone of cells where they're overdividing and they're invasive, these are known as the malignant cancer cells. Okay, so this is now a malignant tumour because we have these malignant cells in it. Okay, and these are the cancer cells. So these orange cells are now cancer cells. They're overdividing and they're invasive. Okay, so the orange cancer cells will invade the surrounding healthy tissue, and moreover they will start invading blood vessels and going into the blood. Okay, and then what ends up happening is they can go all around the body, and then eventually, with a bit of luck for the cancer cell, not for the person, they can end up leaving the blood vessels and going into some new area, and then they'll go into that new area, they'll start over-dividing, and they'll set up secondary tumours, okay? So let me draw this, and the particular area that is really dangerous for this to happen is, of course, the brain. So I'll draw the brain here. So what can now happen is these orange cells can end up in the brain, and they can start forming a tumour in the brain because, of course, they over-divide, because they have that mutation of the first red clone of cells, okay? And this is then known as a secondary tumour. And it's these secondary tumours, specifically in the brain, excuse me, these secondary tumours specifically in the brain that generally kill you in cancer. Okay, and there's another special word for this process of a malignant tumour sending cancer cells into the blood, which then circulate in the blood, leave the blood at some secondary site and establish secondary tumours. This entire process is known as metastasis. Okay, and you would say that the cancer cells have the property of being metastatic. Okay, they can participate in metastasis. Okay, right. So that is a basic picture of carcinogenesis. Now, let me just exaggerate the point that this model is completely oversimplified. Okay, so I have simplified down cancer as much as I can, where we have simplified it to 2 mutations. Okay, we have our original healthy cell that got an original mutation that caused it to over-divide. This produced a clone of cells that are all over-dividing, and then within that clone, one cell was then unfortunate enough to get another mutation that takes you further along the way to cancer. Now, I should just say that within this clone of rapidly dividing cells, it's not just as though you're going to only have cells getting mutations that can take you further on the way to cancer. In reality, you know, these cells will be undergoing mutations sporadically. Okay, some of them will undergo mutations in really important proteins, maybe some protein that's essential for the function of, let's say, complex 4 in the respiratory train, and that cell will die. Okay, loads of cells will get mutations that cause them to die. Okay, it's just that within this clone of over-proliferating cells, eventually, if you wait long enough, one of them will just happen to get a mutation that then allows it to become motile, and then that's going to make this new orange clone of cells, which is then going to invade into the surrounding tissue, invade into blood vessels, and then form these secondary tumours in the brain, and that's what then kills you. Okay, right. So, the point of this speech, however, was to explain how this is oversimplified. I have simplified it down to 2 mutations, overdivision and then motility. Okay, in reality, the progression from a normal cell to a cancer cell is much more complicated than this. These are the major 2 mutations that you need, but in reality there are loads of other ones that you need as well. So in reality, this is far more steps. I think people estimate that it's usually around 7 steps. Okay, so you get a normal cell, it gets a mutation. Generally, the first mutation is that you get overdivision, so that you produce a large population of cells that are all overdividing. So this generally is the first mutation. But in reality, there's loads of steps in between here and then here. Okay, so then one cell in here will get a further mutation that causes it maybe to overdivide even more, or causes genetic instability, etc. Okay, so in reality, there's formation of loads of intermediate clones, basically. So you'll get one mutation in here which will take you further along the way to cancer. It will make a clone of itself, and then within that clone you'll get another mutation in one of the cells, and then you'll gradually make your way towards this final orange clone of cells. So basically, the final orange clone of cells will have far more mutations in it than just these 2. Okay, this generally is the first mutation that you get to take you to the first clone on its way to becoming cancer cells. And this generally is the last mutation that you get, the ability to become motile, okay, and then invade and metastasize. Right, okay, but that's a nice simplified model of cancer that is all that we need to understand in order to understand this topic. Right, okay, so what I'm now going to talk about is the concepts of inter-tumor heterogeneity, okay, and intra tumor heterogeneity. Okay, and although they are terrible, terrible terms, they are really important concepts. Okay, inter-tumor heterogeneity, awful words. Okay, right, so let's start with inter-tumor heterogeneity. What does this refer to? Now, this is a really, really important concept for talking about personalized cancer medicine. Okay, this means that if you go to two people with, let's say, breast cancer, okay, the tumours that they have are not going to be the same, okay? Well, they're not necessarily going to be the same. The idea here is that the etiology that underlies cancer is very different in different people, okay? You'll notice that I have not told you which gene it was that this original cell got a mutation in that caused it to overproliferate, okay? That's because there is not just one gene that can get a mutation in that leads to overproliferation, okay? There are many different genes that you could use here to cause this first initial step. Okay. In addition, there are most likely many different genes that can cause this if they get a mutation in, the motility. Okay. The motility is much less understood. We don't really have a very good idea at all at the moment of what genes you have to get mutations in to cause the motility of the cells. We have a much better understanding of what genes, if you get a mutation in, cause the cells to overproliferate. Okay, but it's most likely that there are loads of possibilities for how you can go from this to this, and that means that if you look at two different people with breast cancer, the actual cancer cells that you've got here at the end of it, okay, the actual orange cells that have the properties of cancer, i.e. they're overdividing and they're invading the healthy tissue and they're metastasizing, okay, they might have very different mutations that are causing that, okay, and that's the concept of inter-tumor heterogeneity, that basically when you look at tumours from different people, i.e. if you look between different tumours, you find heterogeneity, differences in the genomes, basically. The genomes are not the same. Okay, right, so the genomes of the tumour cells, of the cancer cells here, are not the same if you look between two different tumours from two different people, basically. The pathway that the cells have taken from being healthy to being cancer cells is different in different people. Okay, and that's a really important concept for personalised cancer therapy. Okay, right. Then the other concept that I just want to talk about, because it's one of the major problems of personalised cancer therapy, is intra-tumor heterogeneity. Okay, and we'll come on to why this is a major problem later on, but for now I'm just going to tell you what it is. Okay, so intra means within a tumour rather than between tumours. Okay, within a tumour you have different genomes, basically. Okay, this is referring to the fact that if you actually look at a malignant tumour like the one that we have here, we have actually got these different populations of cells. We've got these orange cells here, which are the actual cancer cells, okay? And then we've got other cells here, the red ones. Now, in reality, when you look at tumours, you find more than just two different clones of cells. You find many, many different clones of cells, and that relates to the fact that, as I say, it's a much, there are more steps in this process than just two. Okay, so you form loads of intermediate clones on your way to the final cancer clone. Okay, right. So basically, within a tumour, you have loads of different cells with different genomes. Now, in fact, I'm going to talk about this I'll talk about why this is a problem for personalised cancer medicine right now. Okay, personalised cancer medicine, it means, or at least part of it means, giving drugs that will specifically kill the cancer cells. There's more to it than that, but part of it at least involves giving drugs that will specifically kill these cancer cells. So looking at these cancer cells and deciding what drugs can specifically kill those. Okay? Now, if we just target the actual cancer cells, then we can wipe out this orange population of cells, but we won't wipe out the other clones of cells that are on their way to becoming the orange cells. Okay? So then when we take the patient off the drug, they've already got these other clones of cells that are already really on their way to becoming the cancer cells, and this is one of the key things that then causes relapse, okay, which means that once you come off the drugs, you get the tumour reoccurring, okay, so it gets better for a while, but then it comes back. And the reason it's coming back is because you've got the cells that are on their way to becoming these, and then you're just waiting then for one of these, in our model, one of these other red cells to undergo the same motility mutation that will then take it to becoming an orange clone cell anyway. Okay, so you don't have to go through the entire process again, you're already on your way to getting it again anyway, and that's why you relapse from cancer. Okay, right, so that's one of the major problems with personalised cancer therapy. Okay, right, so there's intertumour and intratumour heterogeneity. Right, so now let's define personalised cancer medicine. Okay, but before we just do that, let me tell you about classical chemotherapy so that we can contrast the aspiration of personalised cancer medicine to classical chemotherapy. Okay, so the idea of classical chemotherapy was very sort of one-size-fits-all kind of thing. Okay, classical chemotherapy refers to very, very powerful cytotoxic drugs, the ones that generally cause you to lose your hair. Okay, now the reason these drugs work against cancer is that generally the way they work is by stopping cell division. Okay, and we'll see many examples of classical chemotherapy in this video when we come on to talk about some examples of personalised cancer medicine, and I'll show you how we're beginning to get personalised cancer medicine using classical chemotherapy. Okay, that will become clearer later on. Okay, so classical chemotherapy agents then work by decreasing the division of cells. Okay, so stopping cells from dividing, and that is one of obviously the key properties of cancer cells, that they over-divide. In fact, if you were going to say one key property really, well, there are two key properties of cancer cells, which are the two that I've said here, that they divide over, they over-divide, and they also invade and metastasize. Okay, but stopping them from dividing is a very, very good thing to do. Okay, the problem with these drugs is that they also stop all your other cells in your body from dividing as well, and that's why they cause the side effects, notably losing your hair. Okay, right. Now, one of the big questions with classical chemotherapy is, why do some people respond so well to classical chemotherapy, and why do other people not respond so well? So basically, we have loads of people, let's say, with breast cancer, and we give them all the same treatment. Okay? If you do this, so I'm just going to draw a little picture. So we've got loads of people here. Here's one person, here's another person, etc. Okay? And let's say they've all got breast cancer. Okay? And we give them all the same drug treatment. We give them some classical chemotherapeutic agent that hopefully is going to stop the cancer cells from dividing. Now, some people will get an absolutely brilliant result. Their breast cancer will go away fantastically. Okay, other people will not get such a good result. Okay, some people in this group will have absolutely horrendous side effects from the drugs. Okay, whereas other people will have much fewer side effects. Okay, so why is the question? Why does some people respond so much better to this anti-cancer chemotherapy drug than other people, and why do some people get the adverse drug reactions whilst other people don't? Okay, so this is a big question. How do we predict whether a drug is going to be effective for someone? Okay, so the efficacy, and also how do we predict the adverse effects, okay, the side effects that a drug is going to have on someone? Okay, well, what you could say maybe it's just luck, but we prefer to think of it as there is a meaning to this. There is a reason that these drugs work better on some people than others, and that there is a reason that they cause much more severe side effects in some people than the others. And what we would like to be able to do is predict which people the drug is going to work in, and which people it's going to have few side effects in. Okay, so This then is what personalised medicine is all about, or personalised cancer medicine we're specifically looking at. So, what then personalised, in fact I'll say what personalised medicine is. Personalised medicine is an aspiration. The aspiration is this, that we will be able to sequence your genome, okay? So we'll be able to look at your entire genome, okay? And we'll be able to predict how effective a drug is going to be. Now we're specifically looking at personalised cancer medicine. Okay, so we can't just look at your germline genome, we also need to look at the tumour genome. Okay, so in cancer medicine it's made slightly more complicated than other personalised medicine because we need to look not just at your germline genome but also the tumour genome. Now what's the difference between the germline genome and the tumour genome? Okay, well, the germline genome means your genome in all of your healthy cells, so the original genome that you had in this original healthy cell here before it became mutated and on its way to this cancer cell. Okay, so the germline genome basically means the genome that you had when your father's sperm fertilized your mother's egg, that genome that was in that original zygote there, okay? Whereas the tumour genome means what genome do the cancer cells actually have. So if we look at the cancer genome, what's its structure? Okay, right. So the idea then behind personalised cancer medicine is that we should be able to sequence your germline genome and your tumour genome, and the aspiration is that we'd be able to plug this information into a computer, okay, and then it would spit out which drugs will be effective against the tumour and also which drugs will give fewer side effects. And that's to understand which drugs will give fewer side effects, we need to look at the germline genome. So the idea is that from this information we would then be able to select a drug which is going to have efficacy, that be very effective at killing the cancer cells, and then also have few adverse side effects. Okay, right, now obviously we are not there yet, it is an aspiration as of yet. Okay, we can't do this yet. However, there are examples of where we are starting to see personalised cancer medicine coming into being. Okay, so we'll call it there for this video, and in the next video what we'll do is we'll talk about some related terms firstly. So we'll talk about what pharmacogenomics is and what pharmacogenetics is and what targeted cancer therapy is. And then what we'll talk about is a little bit of genetics that we're going to need to have a good understanding of. And then we'll go on to the examples of where personalised cancer medicine is starting to come into being.